in this class today, we'll understand the floral biology of legumes in general, and in particular mung bean. So in this, we'll understand the crossing and pollination techniques, and we'll understand why we will do it. Coming to the mung bean, the botanical name of mung bean is Vigna radiata, and the origin is India and Central Asia. That's the reason why we have got a lot of variability in mung bean in our Central Asia and Indian subcontinent. And synonym is, we call it as mung, and in other parts of country has got its local names for this. And basically we use mung bean seeds as a whole grain, or we use it for the sprouts or the split dals, either the dehusked or husked one, as per the preparation of many food items. Coming to the genome size, the mung bean has got the small genome size around 494 to 580 MB, depending on the different species in this, how genotyping has been done of the genome of mung bean. Means very small, around 500 MB on an average. And the diploid chromosome number is 2n22, and it is a self-pollinated crop. Most of the legumes, they are self-pollinated crops. And in case of pigeon pea and other thing, they are a little bit of often cross-pollinated. Okay, that is the reason why the net and other thing they use for maintenance of purity. And coming to the family of this mung bean, it has Fabaceae family of genus Vigna and subgenus Creotropis and section Creotropis. And in this, Vigna radiata and Vigna sublobata has been the classified. And coming to the different wild Vigna species or the different cultivated Vigna species, probably you people have seen mung bean, Vigna radiata, right? And how many have seen black gram? Satish, you have seen black gram?
Yes, sir.
The black gram seed coat color of the this will be a black, and the split dal color is white one. And there is another the wild species of Vigna acunitifolia. This is called as moth bean. In most of the parts of the Rajasthan and arid this one regions, it is cultivated moth bean, very nutritionally rich. Vigna acunitifolia. The grain size is of little bit smaller compared to mung bean and black gram. And there is another the Vigna species that is called as Vigna umbellata. This we call as a rice bean. And this is a very important Vigna species. It has got the MOMB resistance, and it is crossable with the Vigna radiata. And most of the Punjab and other parts, the rice bean has been commercially cultivated, and people eat these rice beans. The size of the rice beans is smaller than the mung bean and black gram, and little bit shape is little elongated compared to Vigna, this black gram and green. And there is another species, Azuki beans. This is Vigna angularis. It is a little bit bolder than the both black gram and green gram. And another species is Vigna anguculata, cow pea. Most commonly, almost all the people have consumed this cow pea. It is used as a vegetable in most parts of the India. And there is another species called as tuber cow pea. It is not much seen in case of India. In other parts of the Asian countries, the tuber cow pea is used. And there is another species that is called as bambara nut. So this is also Vigna species, is very less commonly seen in India. And coming to the inflorescence of the mung bean, we call it as a raceme. And this is a typical peduncle, and in a group in the rachis, we'll get 9 to 10 flowers in a group. This is how what you mean to say, the flowers or inflorescence of the mung bean is observed in on the crop of the mung bean. And this is a floral biology. If we look into the floral biology, the typical mung bean flower it contains 3 kind of petals. First one is standard petal, second one is wing petal, and third is the keel petal. And keel petals have joint. Both the keel petals have joint. And though all things, they are supported by the sepals near the stalk. And then coming to the male and female parts, there is a stigma. Stigma is attached to the style, and there is a ovary inside the this pod, and then there are ovules, that becomes the seeds, and there are stamens. There is a anther and there is a filament of that one, or the male parts in this flower. And this, just look into the inflorescence. This is how an old flower and the young flowers look into. And the flower which is opened like this is the older flower. We won't use it for the emasculation. We use the flowers which are not yet opened. This flowers we use for the emasculation. People understood this one? Why we don't use the older flowers or the open flowers? Yes, Udita? Yes, Udita, why we don't use the older flowers for the crossing program? So coming to the inflorescence in this, as I described you people, 8 to 10 flowers in a group that comes under the inflorescence, and the older the flowers. Why we don't use the older flowers for the crossing program? Because when it opens, it's a clistogamous flower, where it means the fertilization will be over before opening of the flower, and there will be a fertilized flower. This is basically open flower is a fertilized flower, so we won't use a fertilized flower for the crossing program, and we use the fresh young one which is not opened. We open with respect to the use of forceps, and then we remove the anthers. So this is how an inflorescence, as I was describing. And this corolla is of papilionoideous nature. Petals have got 5, sepals are 5 in this, and this is how the keel petal looks. And these are the stamens and anthers here, reproductive organs, and this is the pedicel, and this is a wing flower, and there is a standard flower. And this axillary resumes, it have a flower like this. This is how an open flower looks like this. The wings, standard keel petals. And this is a floral formula how a botanist can describe the mung bean flowers botanically. And if you open all the 5 the petals, take it out. So this is how an stigma looks. Stigma is having the hairy structure like this, trichome like this, and this is a style of the stigma. And then we got a filament and the anthers here, where you will get the pollen here. And all the 9 stamens, usually in this we have got 10 stamens, or 9 are in one, they are grouped, 1 is separate. This condition is called as diadalphous flowers. 9 plus 1 condition we call. and gynoecium is the monocapillary one. This has got a single capil, and stigma is hairy. And why it is hairy? Because when pollen will be in the air or when pollen bursts, so the stigma should be receptive. It can catch the pollen. This is how wing flower looks like this, the open wing petal, and standard or the petal looks like this, and there is a keel petal looks like this when we open the flower. And this is a closer look of diadelphous stamen. So diadelphous stamen, this looks like this. So you have to please closely look at this flower. These all 9 stamens is a united stamen, means fused stamen. All 9 are in a single group, 9, and this is +1. This is a pre-stamen 1. Okay, and you look into the nature here, the beauty of the nature. All 9 are here in the back, and 1 is here in the near to stigma. If there any damage by the insect and other thing at the outer protective the wing and keel petal, so that among the 9+1, if it insect eats here, so this can be protected, and if insect eats here, the all 9s can be protected in the nature to provide the pollen to this stigma and have the seeds for the next generation to survive in the nature. This is the beauty of the nature of the Mung Bean flower, or the most of the Leguminaceous flowers, where 9+1 condition is very typical in this. Okay, and how as breeders we use this one? As a breeders, we emasculate, we cut open the flower, we cut open here, and we remove this one, and this 9 anthers. Before the anther bursts, what we do is we select this unopened flower like this, and then we do the emasculation in the evening times after the sunset, and we just remove, we cut open here like this, and we just release the style outside. And what we do is we remove all these 9+1 anthers, and then we leave like that. We do emasculation in the evening time, we leave it open, and what we do is we do pollination in the next day. Next day in the sense before sunset. Before there is a sunset, we come to the field, we take out the pollen, and then we test the pollen here at the stigma. We pollinate by hand with the forceps, pollen here on the stigma. Okay, this is how an crossing program is done in case of Mung Bean. The time is very important. Emasculation time is very important. This is a little bit of I'm showing an example of groundnut flower, where the wing and standard and keel petals have separately shown here in a proper way. And these are 9 stamens, 9+1 condition is typically shown here. And this is the Mung Bean. One more important thing is, okay, we have done the crossing program, and then we come to field, and how to confirm the cross, whether we have selected plant A and plant B, and we have crossed, whether the F1 is it's a true F1 or whether it's a false F1. How to identify that one? What are the different methods? In case of Mung Bean, it's a very easy. Here you can see the Mung Bean pods. When these pods mature, they turn into black color, and the length of the pod, if it is fully having the full length, means having around 9 to 12 or the 13 seeds in the pods, generally it means it's a self-pollination. Means we didn't do our emasculation properly. While emasculating, there is a pollen anther burst, and there is the chance of contamination or the selfing by the pollen to the stamens. This is how we, if the pod length of the F1 pod length is very longer, then we determine here in the field itself that this is the selfed one. It's not a crossed pod. Then if it is a pod is very small, having the 1 or 2 seeds and a very small 1 or 2 centimeter of having the length, that pod is considered as, or the seed is considered as the crossed one. Typically it is very small, having a 1, or if it is a 1 is the best one. If it is a 2 seeds inside, that also we consider. If 2 to 3 seeds, very small one, we feel that probably that is the crossed pod. And then what we do is we plant those crossed pods in the field along with the P1 and P2 in between F1. Parent 1, parent 2, and in between there is a F1. What we do is we grow them, and when there is before flowering, the field in the plant itself speaks whether it's the self or the crossed one. If it is a cross, if there is the recombination, it is having a mixture of both male and female parent. Morphologically, it is a hybrid like that. If parents are very contrasting, suppose P1 is having the very dark leaf color and P2 is having the very light foliage color, so easily it can be observed in the field. And then among that, whichever resembles the P1 we use at a female parent. If it looks like female parent, it means it is a self parent. We'll discard that plant on the field itself. So this is how in the field we screen for the true F1 or not. And then coming to the if both the parents P1 and P2 looks similar, virtually there is no difference between P1 and P2, because the difference is with respect to the disease resistance or the insect resistance or the yield level. So we can't understand till the pod formation and other. So what should we do? So in that situation, what we do is we'll identify the specific markers for the P1, specific markers for the P2, and then we raise the F1, we extract the DNA in F1, and then we run the markers for P1, P2, and F1. If both the bands in P1, P2 they are coming in the F1, so then at the molecular level we consider it as a hybrid. These are the different 2 to 3 techniques what we use in the Mung Bean. This is how when crossing program is done in Mung Bean, and this is how we'll confirm the. And this is a typical example of Mung Bean crossing with rice bean. So F1 looks like in between the rice bean and Mung Bean. So typical leaf is of, some leaves are like Mung Bean, some leaves are like rice bean, and the nature of the flowering habit comes of rice bean. So this is how it looks typically in case of when we cross between Mung Bean and rice bean. But the problem here is there is no endosperm formation in the F1 seeds. So F1 seeds have very less this endosperm for the supportive nutritive tissue. So seeds are very shriveled, and the F1 is sterile. I mean to say there won't be any pollen in this plant. So what we do is we double the chromosome of this one we take we do the tissue culture here. We use we double the chromosome with the colchicine treatment, and then the double tetraploid is not sterile, and then that gives the pollen, and we'll get the pot set on that. So that we use it for the breeding program. Since the rice bean is having a good source of MOME resistance, so that's the reason why most of the breeders they are using this rice bean into the crossing program to transfer the MOME resistance. So this is the typical class today with respect to our floral biology of Mung Bean, how it is typically used in the for breeding program. Any doubts for you people?
इससे अपनी एमआई रेजिस्टेंस कुछ नाम लिए तो ये मुझे समझ नहीं आई, मतलब क्या है ये चीज?
What is that?
Sir, Monica is here. Sir, MI resistance, MIMR, something you have told.
Yeah, that is Mung Bean yellow mosaic virus. So this is the disease that is very problematic in the Mung Bean. So that is the major thing, major issue for the breeders. This is the one of the thing what we use. Any doubt in this? The Mung Bean yellow mosaic virus is the very notorious virus, and this Delhi is the hotspot for the Mung Bean Mung Bean yellow mosaic virus. When virus hits, it's transmitted by whiteflies, and then completely plant turns yellow, and the pods will be yellow. There is no seeds inside, and then completely the plant dies. So the Delhi is hotspot for the Mung Bean yellow mosaic virus. So let me show you people the MYMV. So MYM is a typical problem here in the in case of Delhi. So this is a major problem. So we are trying our level best to breed for the varieties for this, but this is how and it looks. So I will just try to share the screen where you can see the is my visible? So you just look into this picture. In this is resistant one. This is how it looks like a green. And then comes to the moderately susceptible, and then this is the highly susceptible. How it looks, susceptible and highly susceptible for the yellow Mung Bean yellow mosaic.
इधर होगा, देखो ना, इस दरिया में देख रहे हैं कैंची जहाँ रखा हो.
Yes, you've seen this one, yellow mosaic virus. Are the slides visible? This is visible for you people?
Yes, sir.
Yes, sir.
Yes, sir. Yes.
Okay, this is a highly susceptible MRME plant. So our major emphasis will be on breeding for this one. And yield is the ultimate thing what we concentrate. Either we go for the biotic or abiotic stresses. Ultimately, we concentrate on the yield. So high yielding is the very important trait for the breeding. Okay. Any doubts in this? So that's all the class for the practical class with respect to the Mung Bean, how crossing and selfing will be done in case of Mung Bean, how the flower condition is there, the floral biology. So many of the things will come in exam. So please try to remember the floral biology and the timings, when we do the emasculation and when we do the pollination. Okay, that's all from my. Any doubts?
Yes.
Sir, pollination should be done next day, means before sunset, before sunrise, sorry.
Yes, the pollination should be done before sunrise. Because what happens in the morning, there will be some morning dew. So on the anthers, for example, on the stamen or the on the stigma, there will be some kind of moisture layer. So when we do the pollination with the morning time, so before the sunset, there is a proper dehiscence of the anthers, and there is the pollen availability is very high.
And sir, why is the hybrid pot size small?
See, when we do the emasculation, we'll be damaging the, we are cutting out the wing and keel petal. See, when we do the emasculation, we'll cut it out. We damage the flower. So when we damage the flower, see, it is an unnatural process. So many times, when we do such kind of emasculation, so when we do usually 100 flowers, okay, out of 100 flowers, 3 to 5 flowers will retain on the plant. Remaining 96 or 99, many times 100 flowers will fall off from the plant because we are disturbing the natural flower. This one we are cutting out. So that's the reason why. So the crossing efficiency in case of mung bean is mung bean and lentil, they are very, very less, 3 to 4%. If you do hundreds of flowers, hardly you will get 2 to 3 or 4 flowers. So that is a major problem in case of mung bean. Bridges, did you understood?
Because of the stress, size decreases.
Yeah, because we'll be cutting it. So there is a natural stress. It understands plant takes, because there is some insect damage, some biotic damage. So it tries to remove that flower. It's a natural way of eliminating the insects. Okay.
Okay, sir.
But sir, generally in case of the cross, there must be heterosis, no?
No, yes. Why we are doing why we are crossing heterosis? See, if we get one seed from one seed, we plant in the field. One F1 seed will give us 400 to 500 seeds of F2, and then we plant it, go for F6, F7 in the field. It gives thousands of progenies. From that we do the selection, and that will come as a variety. What's this?
Okay, it means that this stress is only up to F1.
Yes. It's a human created stress.
Okay.
We just want to exploit it.
Okay, got it. Okay, sir, got it.
Okay. It's very simple. Yes, Garima? Any doubt?
No, sir.
Olivia?
No, sir.
Did you understood? Did you follow the lecture?
Yes, sir.
Okay, the major emphasis will be on the how the emasculation, the 9+1 condition, diadalphos, and then how we'll emasculate, and then how we'll pollinate, when we'll pollinate and when we'll emasculate. That is important. And whatever I have shown in the slides, so that please go through those contents. For example, genome size in case of mung bean, the chromosome number, the diploid chromosome number, 2n is equal to 22. And then what are the different wild species of mung bean? So those things are the important for the exam. Okay, Udita?
Yes, sir.
So why we select young flowers, not the old one?
Sir, actually, I missed that point.
Yes, I was asking the same point to you. I asked the same question to you. While I was
Actually, I was in metro, so there were some network issues. I couldn't hear you properly at that time.
okay,
Sorry.
I pointed that to you why there is an why use the young flowers, not the older one for the crossing program. Okay, Priya Upadhyay,
Yes, sir.
why we use old? Why won't you we no? Why we don't use older flowers?
Sir, because they are means
They're old.
they the old flowers are fertilized and
Yes, absolutely. So you have followed the lecture. That's good.
thank you, sir.
So that is the important point. And what is the 2n chromosome number here? So you didn't
Sir.
follow that point, huh?
Yes, sir. Sorry, sir.
Okay, so you please, I'll be uploading this lecture in the YouTube, so you can just look into the many points and the discussion there. So the many of your doubts will be clear. Okay.